into a longstanding interest in characterizing the function of the system in health, but also what has happened so rapidly that there have been speculations and hypotheses what disruptions of the system may play in various diseases. I'll come back to this kind of slide. It was one of the slides in my book, The Mind-Gut Connection. It came out in 2016, which has really changed my own view of this field completely. Two things I want to emphasize here is you see several arrows going in bidirectional manner. So one is that this is really a system that we're talking about, not anything linear between the gut and the brain, or the brain and the gut, or between individual microbes and the brain. It's a system, a complex system with feedback loops that are both contained within the body. So between the gut and the immune system and the immune system in the brain, but also that includes a loop that's the environment. And that's particularly interesting because it has both the environment has an influence on this brain-gut microbiome system in terms of psychological stress or perturbation, early adversity, social support, but also now with the microbiome, we know it has a very profound effect on our health through the microbiome, which then translates this into gut health and systemic health. So I think this is a main message I'm trying to get across. It's very difficult to study this system with the traditional linear understanding and reductionistic understanding, and there's been many problems in translating results obtained, for example, in test tubes or even in really limited animal models into the complexity of this system that we humans are embedded in. So I put a few take-home points in the beginning, and we'll obviously come back to this. So communication between the gut microbiome and the brain is a ubiquitous, ancient, evolutionary conserved system that modulates host behavior. And I'll show you an example for that. So this is not something that only we humans have, but because if you look in evolution, this has been around for a long time and obviously has proved itself to be quite adaptive for the host. This communication connects the mind, the gut, food, and the environment in a way that can best be understood in a systems biological way. So very different from the traditional way that this has been studied or is still being studied in conventional scientific experiments that deal with, for example, gut function or, so in my own specialty, systems biology is not yet the dominant way of studying, functioning and diseases of the GI tract. Then components of plant-based diets act through the gut microbiome to produce gut and brain health. This is a very important thing. This has really changed the trajectory of my career from not being trained at all in nutrition or diet aspects. But once you get into the microbiome, you realize that the main function is really in translating information that's contained in the things that we eat in our diet into molecules that then interact with the gut-based immune system and throughout the body with all the other organs. I'll come back to this at the end. We won't have much time to get into that topic. So the soil microbiome, this has been studied for longer than the gut microbiome, but we don't really know that much more about it. But it has a major influence on the health of plant and gut microbiome and brain health. So there's a growing interest in this whole concept of how the health and diversity and the richness of the microbes that live in the soil influence the rhizome or the root system of the plant, interacts closely with it, just like our microbes interact with our gut, and that these influences then are translated in the plant into chemicals phytochemicals, phytonutrients that travel in the plant from the root system to the fruits and the leaves to protect the plant against stress and pests and all sorts of other perturbations. And then I'm just going to say this here. This is something really encouraging, and I hope talking to an audience like today, this has an impact, because definitely it's the younger generation that will have to make that change from our current food system to something that's much more compatible with our own health, but also with planetary health. So consumers are responding to the paradigm shift with food choices that are good for human and environmental health. I'm absolutely convinced this will not happen through the people that are currently in charge of things, of companies and politicians, but it will have to come from children influencing their parents and young people that see the importance of this. Just a quick overview, microbes basically inhabit every epithelial surface in our body. They, the largest number lives in the intestine, the highest density, particularly the large intestine. 100 trillion has been estimated. What you see here is on the left upper corner. It's a fiber molecule in the gut that's surrounded by microbes, which feed on these fiber molecules. We have another microbiome, very distinct from the gut microbiome in the mouth, plays a major role in tooth health, but influences also functions, cardiovascular health, and probably also the fetus in pregnant women. The stomach, we've known this for a while. There are bacteria living in the stomach that have their own niche and do very different things from the ones that live in the colon and intestine. And you can also see, even in the belly button, we have a unique microbiome. What's important that all these microbiomes, even though they're all made up of microbial organisms, they all have their own community structure, their relative abundances, and they're very stable in their individual habitats. So the one in the mouth doesn't mix with the one in the stomach or in the intestine, and the one in the belly button doesn't mix, for example, with the microbes that are on your facial skin. So they have their habitats on different areas of the body. I mentioned that earlier, that microbes have been, and this interaction between microbes and the nervous system has been around for a long time, which really explains why they are able, these invisible microorganisms are able to communicate with our brain. You can see here they've been around for, the microbes have been around for about 3.5 billion years, 4 billion years. The first 3.5 billion living in the ocean, biggest biomass on the planet, these microbes in the ocean. And they've had a long time, obviously, to perfect a way of communicating with each other and responding to environmental influences in the water, in the oceans. And during that time, they have accumulated a vast number of genes. I'll show you this in a minute, and are able to synthesize and produce things that we only understand a very small percentage of at this point. But what happened about 500 million years ago, some microorganisms, algae at the time, settled in a particular marine animal, the hydra, which is essentially a floating digestive tract with an opening in front and an opening at the end. And this floating digestive tube is surrounded by a nerve net that regulates its movements and its food intake and expulsion. So some microbes were able to settle inside of this floating intestine and interacted over 500 million years with these nerve cells. There were also endocrine cells inside of these animals, exchanged genetic material, and ultimately these nerve nets of the hydra turned into our enteric nervous system. So the nervous system that surrounds our gut and regulates peristalsis and secretion and blood flow in the gut, and ultimately was the basis for development of a central nervous system. So the blueprints for these neurotransmitters, first in the enteric nervous system, but then in the brain, were really developed by these microbes in the oceans over a very long period of time. And this sort of gives you just an impression of the magnitude of this. So humans, as we all know, have about 20,000 genes. There's about 2 to 20 million microbial genes. And as I said, only a very small amount is known what their function is and what they encode. And what's also remarkable that, you know, when we write our textbooks and practice medicine, we essentially neglect 99% of the information that's contained in what's called a holobiont, so the combination of our genes with the microbial genes. Shows you here the red outline of this figure in the middle is our own genes. All the rest is microbial genes. So maybe it's the reason that we understand certain things, like the simple things, the mechanical things, like the pump, the heart, that's a mechanical pump essentially, but that we understand very little about the brain, and that we have very little ways of influencing, you know, brain function, immune function. And so maybe that's the reason, because we really haven't really considered all the inputs that go into the system. Now, these genes and the metabolites that they encode is shown here. So in the middle, you see these, it's been estimated, you know, hundreds of thousands of these metabolites that are generated from food, from things that we ingest. And these metabolites interact with each other. And they basically, some stay in the gut and others influence the immune system, influence the brain, all our organs. The input to the system is the food. That's the main nutrition for the microbes. They thrive on breaking down large molecules contained like in plant fiber, but also many other food components. To figure out how the system operates, obviously, is a huge challenge. There's tens of thousands of phytochemicals contained in our diet, shown here for the Mediterranean diet, which is obviously what microbes love. There's trillions of microbes, hundreds of thousands of metabolites. And you can imagine how difficult it is, or will be, or has been, to figure out how the system operates and how we potentially can correct it with interventions. A simple model that has been created based on what we knew about a couple of years ago is that it's just another way of showing this brain-gut microbiome system, these bidirectional interactions between the brain, the gut, the microbes, in a way that each of these nodes in the system can influence the other. We've known a lot about the molecules in the intestine. So this is what I've been studying in my career. And we thought about 10 years ago that we pretty much knew everything to explain gut function with immune cells and the enteric neurons and neuroendocrine cells in the gut. But we had made all our models without the gut microbes and the input they have on that system. So this is very simplistic to try to have some framework of how we could approach the interactions. Now, in the meantime, a lot of work has been done. This is an exponentially growing field. And the initial focus has been on using the reductionistic, Western scientific model, very successful for certain things, but probably less successful in this case, to identify individual microbes, like you see in the left, these lactobacillus strains or bifidobacterium strains that interact with cells in the gut lining with these epithelial cells and lead to an increase in a certain host hormone or substance, so oxytocin, GABA, brain-derived neurotrophic factor, which then either get into the systemic circulation or act on the vagus nerve, the main sensory pathway between the gut and the brain. And then you can see on the right side, and we won't have to get into this now, based on studies in mouse models, a lot of changes in response to some of these organisms has been demonstrated. So from neuronal plasticity, social behavior, stress responsiveness, anxiety and depression-like behavior. And so that, social function, anxiety and stress-related and stress responsiveness sort of been the major things that have been characterized and linked with neuroplastic changes in the central nervous system. So the next step is obviously the microbes themselves are not causing all these changes in the host. It's what they produce, and there's 3 categories of what they produce. The bottom are diet-derived metabolites, so from amino acids, from complex carbohydrates, polysaccharides, and from polyphenols that microbes break down these molecules, metabolize these molecules, which then lead, if you go to the right side, neurotransmitters and a variety of neuroactive substances such as indoles and phenols, short-chain fatty acids, estrogens, simple phenols. So many of the functions of these end results, what the microbes do to these food components, we don't know yet, but the ones that are shown here, several of them have been implicated now in health and disease states. Another category, if you go one up, you see these host-derived molecules that are input to the microbes. So bile acids, primary bile acids produced in the liver are metabolized by certain microbes, turn into secondary bile acids. And these secondary bile acids then get into the systemic circulation and have been implicated in, for example, cognitive decline and Alzheimer's disease. Something similar happens, like estrogens synthesized by the host, then secreted with the bile into the intestinal lumen. Microbes metabolize these estrogen molecules, and then they're being reabsorbed in the circulation and exert an effect on different organs. So there are these cycles of host-produced substances secreted into the gut with bile, metabolized by microbes, and then they go back into the systemic circulation. And then there's also some, what says de novo here on the top, some metabolites that are generated from biosynthetic precursors, and they produce, they can result in toxins or other, microbial specific features. So now we know, we have the initial phase of the investigations has focused on the microbes, particular microbes, and what they do to animal behavior. Another step has been to identify the metabolites that certain microbes or groups of microbes produce that mediate this effect on the host. And then we still have to ask ourselves, how do these signals generated by the microbes in the gut, in this anaerobic dark world that we never have any view of, how do they get to the brain and exert their effect on our most complex organ, the brain? One pathway that has probably received most of the attention is the vagus nerve, as I mentioned. So this is the gut, this is the brain of a mouse, obviously. And here it's shown how some molecules that are under normal conditions generated by the microbes, how they act on these vagal sensory nerves and are transmitted to the brainstem and influence all kinds of behavior and emotional behavior, and how if this metabolism is altered, for example, in the case of tryptophan, I'll come back to this in more detail, then a different metabolite is signaling to the brain and has been implicated in various disease states like increased anxiety, depression, but also in humans in autism spectrum and cognitive decline. Another mechanism is shown here. So here is the gut again, the epithelial cells, and interspersed are these cells called the enterochromaffin cells that contain 95% of the body's serotonin. The microbes have a big influence on this serotonin production, mainly because they can produce short-chain fatty acids from plant carbohydrates, amino acid metabolites, these tryptophan metabolites, and secondary bile acids. All of these have been shown to act on receptors of these enterochromaffin cells to stimulate serotonin production, but also stimulate release. Serotonin is being released, acts on vagal sensory nerve endings, and goes back to the brain. So this mechanism, I'll show you this in a little bit more detail, is one that we understand quite a bit. So here again, the brain, the gut, these serotonin-containing cells, and again, a bidirectional interaction of the certain microbes with these cells to stimulate an enzyme that is rate limiting for serotonin production, which then these cells have a unique connection, these neuropods connection with vagal afferent nerves, and signal back into the brain. But serotonin is also released from these cells into the gut lumen and influences the behavior of many of these microbes. So again, it's a very integrated, complex system with bidirectional feedback loops. But we have, so a lot of this work has been done by Elaine Shao at UCLA in her laboratory. Now, tryptophan, this is the essential amino acid that we ingest. So one pathway is the serotonin pathway. Another one is this unpronounceable word, kynurenine, which is also modulated. This pathway is modulated by microbes, by lactobacilli. So the abundance of lactobacilli determines how much of tryptophan is converted into kynurenine and how much into serotonin. So if you have a decreased number of these lactobacilli, there's decreased inhibition of this conversion, get a higher level of kynurenine. And kynurenine has been shown in animal models to have a negative effect on brain function and neuroplasticity and neuroinflammation. And then there's this third pathway, which ends up with the indoles. And this is totally dependent on gut microbes. It's not just modulated, but the microbes are the only ones that have the enzymes that can convert tryptophan into indoles. And many of these indole metabolites, again, have been implicated now in Alzheimer's disease and autism spectrum disorders. So this shows you just for one molecule, tryptophan, how the microbes play this crucial role in determining how this is shunted, how these metabolites are shunted to different pathways. And the balance within these pathways ultimately determines what effect this has on the host, on health, and what's changed in disease. Another communication pathway from the gut to the brain is through the immune system. So there can be, in the normal situation, there's very little activation of the immune system. And this is partially affected by short-chain fatty acids that have an anti-inflammatory effect. In in a dysbiotic gut, so with an imbalance of the gut microbes, there's increased immune activation at the gut level, but also some of the signaling molecules like lipopolysaccharide, which is the cell wall of certain microbes, can get into the systemic circulation and cause a systemic immune response, which then ultimately affects immune cells in the brain and can lead to a chronic neuroinflammation. Again, in terms of cognitive decline in Alzheimer's disease, is a sort of an important mechanism that is being studied. I'll come back to this at the end when we talk about diet. So an interesting group of molecules are these polyphenols. So these are, you may have heard about these polyphenols, but are not really known that this is the chemical name for them, because they're always mislabeled as antioxidants, which they are if you test them in a test tube, but not if you feed them to an animal or if you eat these polyphenols with your plant-based food. So they're such large molecules that they cannot be absorbed in the small intestine intact. They travel throughout the gut down into the large intestine, where they're being broken down by microbes into much smaller molecules and smaller phenols. So some of these molecules are shown here, some of these metabolites. And these smaller molecules are not only able to cross the gut epithelial barrier, but also the blood-brain barrier, and can have a protective effect on the development of these neurodegenerative changes that come normally with age. So a high intake of these polyphenols and a conversion of these polyphenols into these small molecules can lead to a suppression of neurodegenerative changes within the brain, resulting in healthy aging of the brain. This is an area of significant interest, not just in general, but our lab has sort of several studies now that look at these mechanisms. So based on some of the things that I've shown you, and mainly on results obtained in animal models, the several speculations have been made early on in review articles, and this has sort of driven a frenzy of media attention. So people have postulated that they have neurological mouse models that are characterized by an altered microbiome. There's a list of all of these where this has been shown. This was sort of followed by another stage of studies, more relevant, not just the association of these mouse models with these altered disease states, but that also, for example, studying animals that don't have any gut microbiome, germ-free animals, and then introducing microbes from patients, for example, or suppressing the microbes with antibiotics, enhancing them with probiotics, or just looking at what happens in these animals that have no microbes in their gut. This has sort of further confirmed this concept that many of these human brain disorders have a relationship with an alteration in the gut microbiome. So these are all animal studies, and with all the limitations that mouse models have for translational relevance. And so now we're at this level, neurological disorders that have been shown to have, people have studied patient populations, looked at the gut microbiome, and have found that many disease states have alterations in gut microbial composition, in the pattern of metabolites, and the field now is at a stage that these associations, which don't really prove causality, that microbes actually cause the disease, they're just associated with it, But combined with these mouse models where you can manipulate the microbes and see what happens, particularly transplanting stool from patients, transplanting the stool into the gut of these mice and see what their behavioral changes are. This is where the field is now, and still, generates a lot of enthusiasm, a lot of optimism that we are getting closer to understanding some of that system. So we've talked a lot about what what microbial species and strains have been implicated. Haven't really emphasized enough that one of the most unique interactions of these microbial strains and their metabolites is with the gut-associated immune system. So just a few microns under the surface, away from the bacteria, is this immune system in the gut, which makes up about more than 50% of our entire immune system. And it's been shown that gut microbes and their metabolites can influence some of these immune cells and result in brain inflammatory states and all these behavioral states. So microbial immune interactions is probably the initial step to understand what happens in the rest of the body. Now, the microbes and their metabolites, it's not a static system. So this clearly undergoes a development early on in life, primarily the first 2 or 3 years of life. So, animals and humans are born with virtually none of their own microbes. The first inoculation comes when the newborn goes through the birth canal and gets a vaginal fecal imprint from the mother. And then there's a lot of factors that have been characterized in these first 3 years of development of the system that have a major influence. So one is the birth. I was saying, the birth canal is a major way of starting this development. An increasing number of women undergo C-sections, so this initial contact is no longer the case, and it's been shown that the gut microbiome of newborns can be very different between a C-section and a vaginal delivery. Then there's also a lot of things happen before birth, a chronic disease, a maternal infection, particular maternal nutrition status, maternal stress, all have been shown to influence the development of the microbiome in the infant. And then, breastfeeding versus bottle feeding, and other environmental influences. So these first 3 years of life see a tremendous shaping of the gut microbiome. And it's sort of, when you look at it from an evolutionary standpoint, it's basically programming the gut microbiome for the world that this infant is born into. So nature estimates, based on all these factors, what the life of this organism is going to be like, and it programs it in the best way possible. This may have worked in ancient times, but it certainly is no longer working in our modern times, where all these factors are heavily influenced by lifestyle factors or things that have even happened in the last 75 years in terms of nutrition, maternal obesity, metabolic syndrome, breastfeeding, the birth, the mode of delivery, and environmental factors. I think I'm going to skip over this. Now, in the adult life, so on the previous one, I showed you what happens in the programming phase. In the adult life, there's essentially 3 factors. They play a major role in modulating how this interface and its communication to the brain operates. One is the medications we take. Antibiotics are on top of the list. It's horrendous, the number of courses of antibiotics that an infant in the US receives by the age of 2, I think it's 10 courses of antibiotic treatments in a phase where the programming goes on, but throughout life as well. We take antibiotics for colds and all kinds of viral infections without really an indication that this may be doing anything good. Nutrition becomes a major factor that modulates this early programmed microbiome. And then the brain, through stress and emotions, has another major influence on the system throughout life. So based on these developmental phases, I think we can say that there's 3 types of disorders. Developmental disorders that probably start early on in life when the microbiome is programmed. And this is like things like irritable bowel syndrome, anxiety, depression, ingestive behavior, autism spectrum disorders. They all start possibly even during delivery, the influences that the infant receives during that phase. Metabolic disorders, as you know, we are living in an epidemic as bad as our current pandemic in terms of impact, an epidemic of metabolic disorders, obesity, and metabolic syndrome, which all have consequences on secondary diseases, cardiovascular, cancer. And then we also see a growing number of these neurodegenerative disorders, partially because our populations are getting older, but it's a wrong concept to assume just because people get older, they have to develop Alzheimer's or Parkinson's disease. It's really the combination of these early influences. And if you're exposed to that for a lifetime, what effect this has on brain function and brain inflammation. So I think we should probably go a couple more minutes with the presentation, then that we have enough time for the discussion. So just a few comments of what I've told you so far. Hopefully some of these things have stayed with you. The predictions from this systems view of brain-gut microbiome interactions. So once the system is established, it's fairly stable and resistant and resilient to perturbations. That's a good thing. So we don't change our personality every time we eat lunch or dinner, or when we take a course of antibiotics or have a gastroenteritis. On the other hand, once we have a major change in our gut microbiome, it's a problem because it's very difficult to change it back to normal. So it's a good and a bad thing that this system is resistant and resilient. And as I said, most individual perturbations in the adult have a relatively small effect on system output. It is remarkable considering how much our diet alone has changed, and the chemicals we ingest from commercially grown food and all the medications that we take, that we're not all that our bodies are not completely out of control. It is because most of these perturbations, the system is resilient and maintains its basic function even with these perturbations. As I said, the negative side is that most therapeutic interventions have small and transient effects. And one example that always comes up is fecal microbial transplantation. So why not just transplant the stool from a healthy donor who has no problems, health issues, into somebody who has a compromised microbiome, either because of early life experiences or a disease state or really bad diet. The problem is it only works in one single condition so far, and it works really well in that condition. That's a particular type of colitis, or colonic inflammation, called C. difficile colitis, where it's curative, such a transplant. But it's been tried in all the other diseases, from inflammatory bowel disease to obesity to metabolic syndrome, and unfortunately has not been successful for these reasons that I mentioned. Now, let me see. I think I'm just going to quickly go through this. There's a lot of really interesting things that we could talk about, but let me say, and then I'm going to end with one more slide on this. So this whole issue, I mentioned this before about the important role of a healthy diet on gut function and beyond. So on the left side, you see schematic depiction of a healthy gut, the microbes separated by a mucus layer, a dense mucus layer from the lining of the gut. But you do see in the gut, there are cells, immune cells, like these dendritic cells, that actually have sensors sticking out into this into this mucus layer. So as long as the microbes don't come in contact with this is not activated. But what happens with Western diet and with chronic stress, so Western diet, mainly high fat, low fiber, high sugar, the whole microbial community, the diversity, the richness, relative abundances of certain microbial strains changes dramatically. And we could almost say in 40% of people living in America and eating a Western diet, this is the way their gut would look like, if you could look at it. And the big problem is that now some of these microbes can come in contact with these immune cells and cause a low-grade inflammation in the gut. If this gets worse, then there is a compromised ability of gap junctions between epithelial layers, and the gut becomes leaky. So some microbes actually are able now to get into the immune system layer of the gut, and even some into the systemic circulation, like the lipopolysaccharide, and then can ultimately go to the brain, but also to many other organs. So this is really the main problem that I think we face today with a growing epidemic of obesity and metabolic diseases, that this is not just a cosmetic problem, and it's not just a problem that your cholesterol goes up. So we recently read a statistic that based on current criteria to treat elevated cholesterol and blood lipids, 100% of adult males in the UK would have to be on a statin. So our medical system responds to this situation with basically just covering it up and not dealing with this side, which is a diet-related side aspect, but with medication suppressing then the negative end product and decreasing mortality and morbidity from cardiovascular disease. Let me go to the end. So a lot of what we eat, obviously, other than hydroponics and things that are artificially grown, comes from plants, and the plants have a very close interaction with a very complex system in the ground, including microbes. So depending on how our food is grown, the phytochemicals and phytonutrients in the plants that we eat, they may look beautiful, but they may be deficient in many of these health-promoting molecules. And this is another thought to how industrial agriculture is closely connected to the problems that we have in our gut microbiome, the decreased diversity and richness as a cause of these diseases that I mentioned to you.